Welcome to Inside Drug Discovery. This video describes the discovery program of elironrasib, a cancer treatment that acts through covalent inhibition of KRASG12C on. The program is summarized based on a Drug Annotation article in the Journal of Medicinal Chemistry. The article was authored by chemists at Revolution Medicines in California. A link to the article is available in the video description. Before we start, I'll note one thing. In the same issue of JMedChem, the scientists from the same company put out a second drug annotation article on another KRAS-on inhibitor. So both final compounds have the same target, but their methods of engaging the target are different. One is a covalent inhibitor, that is this video. The other is a reversible inhibitor. The two final compounds have many structural similarities. A link to the second article can be found in the video description if you're interested. Okay, let's get started. RAS, which stands for rat sarcomavirus, is a protein family that helps manage cell growth and differentiation. Mutations in RAS proteins can lead to dysregulation of cell pathways and in some cases cancers. KRAS is a member of the RAS family. A specific KRAS mutation, G12C, is responsible for perhaps over 150,000 new cancer cases annually in the United States. These are often non-small cell lung cancers. KRAS, which has kinase activity, has become a well-recognized drug target for cancer therapies. These compounds are KRAS inhibitors, ideally with selectivity for the mutated form of KRAS as opposed to the properly functioning wild-type KRAS. For its kinase activity, KRAS requires a cofactor, GTP, and KRAS exists in two forms: KRAS off, which is bound to GDP, and KRAS on, which is bound to GTP. KRAS inhibitors generally target the KRAS off form. KRAS on, however, is the active oncogenic form of KRAS, and KRAS on inhibitors may carry additional therapeutic benefits. Unfortunately, the binding pocket for KRAS on is shallow and less druggable. Scientists have learned that KRAS on binds to chaperone proteins such as CypA, and CypA therefore represents a potential opportunity for the design of a KRAS-on inhibitor. Here is a simple scheme that shows the general idea. On the left, we have three species: KRAS-on, a potential inhibitor, and CypA, a chaperone protein. The inhibitor is designed to bind CypA. CypA can then undergo its usual binding to KRAS-on. In the process, the inhibitor is brought into proximity to KRAS-on. The final outcome is an inhibitor sandwich between KRAS-on and CypA. The complete complex, the tri-complex, if stable, then has bound KRAS on, which will be inhibited of its oncogenic properties. To enhance the stability of the tri-complex, the Revolution scientists sought to design an inhibitor that covalently links the inhibitor and KRAS on at the mutated cysteine residue. This is an ambitious plan, but the Revolution scientists had some precedent to build upon. This slide shows a compound that was developed by scientists at Warp Drive Bio, which was acquired by Revolution. The compound has a part that recognizes CypA and a linker to a disulfide group that can exchange with the cysteine thiol of mutated KRAS. The disulfide exchange provides a crosslink to KRAS-on, and we'll refer back to this crosslinking in later slides. The scientists also had X-ray data on the full tri-complex of CypA, the inhibitor, and KRAS-on. With this structural information, the scientists were able to advance from this tool compound to a viable lead. On the screen, we can see some of the early changes to generate compound 5, which enabled proper SAR studies and lead optimization. These changes were greatly informed by X-ray co-crystals and likely molecular modeling. There are two key changes. First, the binding conformation of the original compound allowed the formation of a macrocyclic ring. Parts of the macrocycle enhance binding to CypA. The second change is replacement of the disulfide with an acrylamide for covalent binding to the cysteine residue in KRAS-on. And acrylamides have a strong precedent in other approved covalently binding kinase inhibitors. The scientists started their SAR studies on compound 5 around the aryl group on the 2 position of the indole ring. This part of the molecule binds CypA. Here are selected changes that were explored. Going across the top of the table, we see the biochemical potency, the EC50 in a cellular assay involving H358, a KRASG12C mutant cell line, and the extent of cross-linking between the test compound and KRAS. Compound 5 is the original lead. We have moderate biochemical and cellular potency, as well as cross-linking, fine-tuning of the group on the phenyl ring, and ultimately addition of a nitrogen to form a pyridyl ring gave a boost to both biochemical and especially cellular potency. Note that the bond between the indole and aryl R group is hindered enough to allow isolation and separation of the rotational isomers, the atrope isomers. The S form of 14 was found to be the more active diastereomer and advanced as the new lead. I'm going backwards here, back to compound 5 to recall the Michael acceptor on the right side of the molecule. The previous slide introduced modifications to the 2-aryl group on the far left. Now we're looking at analogs of the N-acylpyrrolidine. All the compounds in the next slide have the optimized 2-aryl group, as well as one other change, removal of the phenolic OH. This table shows just 4 analogs. We have dropped a column for biochemical potency with an emphasis on cellular potency, selectivity, cross-linking, and some indicators of stability. As you can see, progressive modifications around the unsaturated amide markedly boost cellular potency with corresponding gains in selectivity over wild-type KRAS. Blood stability and mouse clearance were also encouraging. Note that blood stability is relevant for Michael acceptors as conjugation with glutathione as a Michael donor is a potential contributor to extrahepatic clearance. Compound S23 was advanced to explore variations around the pyrrolidine ring, which I'll refer to as the linker between the macrocycle and the Michael acceptor. In the paper, the authors reported nearly 10 different linkers. I suspect they tried more. I'm just showing 3. The parent in the series, compound S23 with the pyrrolidine ring, this spirocyclic analog, and finally a piperidine analog, compound S33. Again, the MedChem team managed to achieve further gains in cellular potency. The bottom compound also had reduced clearance and improved oral bioavailability in mice. Its one reported liability was solubility. Solubility at 7.4, pH 7.4 was quite poor and required additional optimization. For this final push, the researchers focused on this aromatic ring in the macrocycle, which was where a phenol was present in some of the early lead series. Again, here are just some selected compounds from the article. Options explored include replacing the aromatic ring with a morpholine and a piperidine ring. The piperidine maintained potency, but the solubility gains in the morpholine analog were deemed more essential. Ultimately, compound S41, with a research designation of RMC6291, balanced the key criteria for additional study. Here are some ADME and PK data. S41 has modest membrane permeability and reasonable stability in human blood, though the half-life in monkeys is quite short at less than an hour. Note that covalent inhibitors, even if they are not detectable in plasma, may still be engaged with the target and exerting an effect. Although S41 is technically a small molecule, it certainly falls beyond the rule of 5. Finally, S41 did not demonstrate any notable safety risks in standard in vitro assays. S41 showed promising in vivo efficacy. The results of several studies can be found in the paper. I'm just showing one, a mouse xenograft study with a KRASG12C mutation. There were 3 dosing groups: untreated, daily oral dosing at 25 mg per kg, and then a 100 mg per kg group. The 3 groups showed clear outcomes. Not one of the animals in the control group showed remission of over 10%. In fact, all animals in the control group showed tumor growth of over 300%. The lower dose group had a mix of regression of up to 80% and regression of over 80%. The high dose group uniformly showed regression of over 80%, with many animals showing complete regression. Based on these and other data, RMC6291 was advanced as a clinical candidate under the name elironrasib. Elironrasib is currently in multiple clinical trials, one of which is shown on this slide, in patients with non-small cell lung cancer bearing the KRASG12C mutation. That completes the story of the discovery of elironrasib. Elironrasib is an oral covalent KRASG12C ON inhibitor that is in early development for the treatment of certain oncology indications. Thank you for watching all the way to the end of this video. I welcome comments and questions. Please like and subscribe. Take care.